So today we will be starting with cell adaptations and we will be completing with its two topics that are atrophy and hypertrophy. Let's start. As we know from the last lecture, a cell can be injured and it can respond to that injury via three phases, that is adaptations, reversible cell injury and irreversible cell injury. So in cellular adaptations, there are different methods to cope up. These methods are hypertrophy, atrophy, hyperplasia, metaplasia, and dysplasia. Atrophy is decrease in size of cell. Hypertrophy is increased size of cell. And hyperplasia is increased number of cells. Whereas on the other hand, metaplasia and dysplasia are way different from the above three. Metaplasia is change in shape or transformation of the cell. And dysplasia is developmental problem of the cell. We will be studying these all topics in quite detail under these headings. First one, what is that condition? Second, what is the cause of that condition, which may be physiologic or pathologic? Then we will see how that organ appears which has gone that condition in two different manners, the gross and the microscopic and the molecular pathology. So let's see the diagram. This diagram is the perfect representation of cellular adaptations. Like you can see, it is decreased cell size, it is increased cell size in the cell. This one cell has divided into three cells in hyperplasia. In metaplasia, it has changed its shape altogether, and in dysplasia, it is something different. So let's start with atrophy. What is atrophy? Like we said, it is decrease in size of cell. Physiologic cause of atrophy is aging process. The most common example of it is atrophy of thymus in adults. And another common example is atrophy of gonads in females post-menopause. Then there are pathologic causes for atrophy as well, which are stated below as starvation atrophy. Starvation atrophy usually leads to weakness in the body and emaciation or very thinness. Did you know how the food that we eat get utilized? So the order of depletion is basically carbs get utilized first, then fats, and then protein. Then the second thing is ischemic atrophy. Ischemic atrophy leads because of when there is less oxygen or less blood supply to that organ, there is less oxygen supply, so that cell does not develop properly in size. Therefore, its size decreases. Then we have disuse atrophy. Disuse atrophy is again pretty self-explanatory, because if you don't use a thing properly or for a long period of time, it is not going to develop properly, right? So when we have a plaster, that limb or that hand is gonna stay fixed for a long period of time, and that cells are not going to work properly, so it will not be growing that fast. Then we have neuropathic atrophy. Neuropathic atrophy is again intervening the nervous system. So when the nervous system gets intervened, the signals does not pass as efficiently as they should, which leads to muscle wasting. The best example of it is poliomyelitis. Then comes endocrine atrophy. Endocrine atrophy usually leads to decreased metabolic activity or like hypothyroidism and hypopituitarism. There are other three also like pressure atrophy, developmental atrophy, and idiopathic atrophy with unknown cause. After this, we lead to the morphologic features of atrophy. that when an organ undergoes atrophy, how does that organ look? What do you think? What is the basic thing comes to your mind? Yes, right. The organ would look small and shrunken. And what about the cells? How would the cells would look if the organ has undergone atrophy? Yes, agreed. Cells would also look small in size, right? But do you think they will die or would they still be alive? They would be alive. Why would they be dead? Because only the size has reduced. They haven't stopped functioning. And one more question. How do you think the cells decrease in size suddenly? Like, is this a miracle happening? Is there a random process? No. So cells decrease in size because of decrease in size of three basic things, which are endoplasmic reticulum, mitochondria, and myofilaments. So when these three things decrease in size, the whole cell decreases in size. Apart from this, there is also increased number of autophagic vacuoles. Autophagic vacuoles is they eat their own stuff. So autophagic vacuoles contain cell debris and they also form residual body in cytoplasm. So this is the morphologic features. Now let's move on to molecular pathology. Like what would happen after the organ has undergone atrophy. There would be imbalance in cellular protein pathway. Why would be there an imbalance? Because there would be decreased synthesis of protein and increased cellular degradation of protein. That means this protein would not be made as fast, and if made, it would be degraded as soon as possible. But how does these things happen? These things happen due to lack of nutrition for synthesis, and for degradation there is activation of something called as ubiquitin ligases. These ubiquitin ligases attach to the cellular protein and cause them to undergo degradation. I will use another highlighter for better clarity. Sorry for the delay, just a second. Yeah. So this was all about atrophy. I hope it is understood. Let's move on to the next topic, hypertrophy. Hypertrophy is just opposite of atrophy. Like basically what it is increase in size of cells. To be specific, parenchymal cells. And it affects all types of muscles. It is not specific to only one type of muscle group. So when stress strikes the cells, when stress strikes non-dividing cells, it undergoes only hypertrophy. That is increase in size of the cells. Non-dividing cells are like cardiac cells, neuron cells, and other things. But when stress occurs in the dividing cells, it occurs not only hypertrophy, but it also undergoes hyperplasia. Usually in dividing cells, hypertrophy and hyperplasia go hand in hand. It's quite common. But what is the cause of hypertrophy? Why do hypertrophy happens in the first hand? So it is usually due to increased functional demand or due to increased hormonal stimulation. Now again, if there are causes, it would be of two types, physiologic and pathologic. The physiologic cause, example of physiologic hypertrophy is increase in size of uterus during pregnancy, as pregnancy is a healthy process. But if it is a natural process, how does hypertrophy occurs? What is the cause of hypertrophy during pregnancy? It is due to estrogenic stimulation. And in pathologic causes, there are four different types how it can be hypertrophied. First, there is cardiovascular diseases. Cardiovascular diseases like systemic hypertension, aortic valve disease, mitral insufficiency, these can cause hypertrophy of cardiac muscles. Then there is smooth muscle hypertrophy. In smooth muscle hypertrophy, there are examples like cardiac achalasia, which occurs in esophagus, and pyloric stenosis, which occurs in stomach. No, I know it is a cardiac, but cardiac is not always related to heart. Remember that. Then we come to skeletal muscle hypertrophy. Skeletal muscle hypertrophy is usually more commonly seen in athletes and manual labor. Like, have you seen those athletes like Usain Bolt who have massive, large legs, or the manual label that picks up the stones and things? Why do they have such big muscles? It's because they have used them continuously and developed them over time. And even so, great extents. That's why their muscles have hypertrophied. Next, we come to compensatory hypertrophy. Compensatory hypertrophy, the word is quite self explanatory. Like, if one thing gets damaged, the other will have to take the whole load of it and work for both of them. Example, post nephrectomy. Post nephrectomy, the one kidney has to take the load of everything and it undergoes hypertrophy and hyperplasia and has to perform the function of both since one has been removed. Then we come to morphologic features. How would the hypertrophied organ look? Just imagine. What comes to your mind? Yes, it would look large and heavy. Microscopically, it would be like increased muscle fibers and increased nuclei. And yeah, also basics. There would be again increased mitochondria, increased myofilaments, increased endoplasmic reticulum, due to which the cells increased and the muscle fibers increased in size. And last but not least, we come to the molecular patho. Molecular patho, again, it is just opposite to atrophy. There is increased protein synthesis in the case of hypertrophy. So, yeah, this is hypertrophy. Let me show you a photo of an hypertrophied heart and how does it look. So, basically, this is the photo of an hypertrophied heart. So, let me give you a context. A normal average human heart weighs around 300 to 350 grams, whereas an hypertrophied heart weighs around 700 to 800 grams. Yeah, I know it's a huge difference, right? So, this is an hypertrophied heart. So, yeah, today we have completed cell adaptations, basics, atrophy, and hypertrophy. I hope you understood what all this is. And in the next lecture, we will do hyperplasia, metaplasia, and dysplasia. Thank you.